Antibiotic-based selection is a very common technique that we use in biochemistry and molecular biology in order to select for bacterial cells that have actually taken in the plasmid that we want. So a plasmid is a circular piece of DNA that can be hosted outside of the bacteria's own chromosomal DNA. And so we can use these plasmids as vectors or vehicles for putting in a gene of interest. And so this might be a gene that you want to have more copies of the DNA made, or you might even want to have the bacteria make the protein that the gene encodes for. So what you do in molecular cloning is you can actually take that piece of DNA that has the instructions that you want and stick it into a plasmid and then stick that plasmid into bacteria. But you want to make sure that you only are going to allow bacteria to survive that have actually taken in the plasmid. So when we put the plasmid into the cells, this is a process called transformation. And it's not completely efficient. So some of the cells might not have taken in the plasmid. We don't want those cells to be able to grow because then we're just feeding them and we're paying basically for these cells to grow when we don't really want them. They're not doing anything for us. And they might even have a fitness advantage because they don't have to copy that plasmid and make the protein and anything like that. So in order to make it so that only the cells with the plasmid, so make it so those cells have the advantage, we use antibiotic-based selection. So in addition to having a gene for the thing that we want, we also have an antibiotic resistance gene on the same plasmid. This way, we can then grow the cells in the presence of the corresponding antibiotic, and only the cells that have taken in the plasmid and thus have the antibiotic resistance gene will be able to survive. So when you're doing a transformation, you plate the cells, and then the cells that have the cells will find a home on the plate, and if they're able to survive, the plate has the antibiotic in them. If they have the antibiotic resistance gene, they'll be able to take root and kind of grow, and they make copies of themselves on top of themselves, and you get these gloopy dots called colonies. A note of caution that this means that the cells have the plasmid. It does not mean that the plasmid actually has the gene that you wanted to put in there. And so then you can use techniques such as colony PCR, analytical digest to see if it's likely in there, and then sequencing to confirm that all the sequence is correct. But the first step is this selection mechanism. And we do this using a variety of different antibiotic and antibiotic resistance pairs. So different plasmids will have different antibiotic resistance genes. And typically when you buy a plasmid, it'll give you information about what antibiotic or antibiotics it has resistance for. It's helpful that we have lots of different pairs of antibiotic and antibiotic resistance genes, because for a number of reasons, including the fact that sometimes you actually want to put in multiple plasmids into a cell. And this way you can use multiple antibiotics to make sure that each of the cells have each of the different plasmids. So you want to make sure that if you're putting multiple plasmids in, they each have a different antibiotic resistance gene, and then you grow in the presence of all of them so that you have only the cells with all of them will be able to survive. This is used, for example, when we're doing things like blue-white screening, where in this case, we actually have multiple plasmids in the cells, and so we use multiple antibiotics to make sure that all those plasmids are there. This is also a good time to note that a difference between screening and selection. So in selection, we're only letting the ones that have something grow. And in screening, we're basically saying, okay, show me the ones that have something and the ones that don't have something. So blue-white screening is this way where we can see if cells likely have an insert in the plasmid because the insert is going to disrupt a product that's needed to make a blue thing. And so if you have the insert in there, then you're going to disrupt the blue product making and then get a white colony. Whereas if you don't, you get a blue colony. But with this selection, here we're actually only the colonies that are there are going to have the plasmid in them. And so you're selecting for the colonies that have the plasmid. I should note that this isn't foolproof. And so sometimes you'll see things like little satellite colonies, and we'll talk about these. This can happen if your antibiotics get degraded or if the antibiotic is actually like shipped out of the cells, which is the case with things like beta-lactam antibiotics like ampicillin. And so we'll get into this, as well as some of the other technical details for and considerations when using these antibiotics. But let's start with a quick note on naming. So antibiotics, these are usually naturally produced by other bacteria to protect themselves from bacteria. And so there are a lot of modified versions of antibiotics, especially because antibiotic resistance is a big problem because bacteria are tricky and they can evolve resistance. And then, so this is the same resistance that we take advantage of in some cases in order to use antibiotic-based selection. But the like original antibiotics and ones that we still use often are modified versions of these. They come from bacteria, and based on which kind of bacteria they come from, there are different naming schemes. So ones made from streptomyces bacteria normally get names ending in mycin, so like streptomycin, kanamycin, and those from micromonospora bacteria get names ending in mycin or MICIN instead of MYCIN, so gentamicin. Yeah, I don't know how to pronounce these things to make them sound different, but that's how I always say them, but the spelling, that's why. So let's get into some examples. So if we're talking about antibiotic-based resistance or antibiotic mechanisms, we need to think about how antibiotics work and how we can prevent them from working. Antibiotics work by taking advantage of things that are different about bacterial cells from our cells. And typically the two main things that are targeted are going to be cell wall synthesis, which is targeted by ampicillin and other beta-lactams, as well as translation, which is targeted by like tetracycline and other tetracyclines, kanamycin, other aminoglycosides, and chloramphenicol, various different antibiotics that we'll talk about. Why do these strategies work? So cell walls, this one is easier to explain. So bacteria have cell walls and we don't. So if we target cell wall synthesis, we can target bacteria without harming our cells. We're going to be able to target translation because the bacteria ribosome, so the protein making machinery, is different enough in bacteria that we can have drugs that bind to bacterial ribosomes and prevent protein production without harming human ribosomes. There are various strategies that cells use in order to inactivate or kick out the antibiotic. So some of the antibiotic resistance genes are going to modify the antibiotic, break them or add something to them that's going to prevent them from binding or prevent them from having their activity. Other antibiotic resistance mechanisms include efflux pumps, so actually pumping out the drug that comes into the cells. And so we'll see examples of both of those types of things. Both the modification with ampicillin resistance, kanamycin resistance, chloramphenicol resistance, as well as pumping out, which we see with like tetracycline. And so I'll show you these examples when we're going through. Let's start with the cell wall synthesis. So bacteria have this cell wall, and it's made out of these peptidoglycans. So these have these so little peptide chains, and glycans, so sugar. And so there's this like sugar linked, these peptide linked sugars. And in order to make those peptide links, the bacteria use an enzyme, so a reaction helper called transpeptidase. What this transpeptidase does is it's going to take these peptide chains and actually attack them to form a temporary bond where it's stuck on the transpeptidase. And then then a second peptide is going to attack that. And so you get this peptide-peptide bond formed, and then this transpeptidase is released. And you get these chains linked, so you have a strengthened cell wall because you're making all these cross links. What happens when you have a beta-lactam antibiotic is that the transpeptidase is going to attack the antibiotic, kind of confusing it for the attack site on the peptide. So what we call these a beta-lactam antibiotics because this lactam is going to be this like strained 4-member ring. And these beta-lactam antibiotics, when they get attacked, they break open. This is going to get them permanently stuck on the transpeptidase. This is an example of a mechanism-based, or like sometimes called like a suicide inhibitor, because it's practically irreversible. Because once it breaks open, it's very, 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 very unlikely that it's going to come off. You can't like reverse this. It's not going to go backwards, because once you relieve this ring strain, now it's way happier. And so it's going to be stuck on there. And when it's stuck on there, this transpeptidase can't do its normal job of building cell walls. So the beta-lactam part is what makes these antibiotics, gives this class their power. And so different beta-lactam antibiotics have different variations on the structure, but they have the same beta-lactam core. So common examples include penicillin, carbenicillin, methicillin, oxacillin, amoxicillin, cephalosporins, monobactams, and carbapenems. And so there's a lot of different penicillin class antibiotics. And as for the structure was actually figured out by Dorothy Crowfoot Hodgkin and Barbara Lowe. And I have a post on this as well. They're really groundbreaking, pioneering X-ray crystallographers. So really, really cool stuff. And this was a really important discovery, figuring out the structure and how it works. And now we have all of these various versions to choose from, especially if bacteria get resistance. So that's how beta-lactam antibiotics work, but how do they not work? How do we make resistance to them? Often this is done with this like BLA gene, which encodes for beta-lactamase. So when you see A's, think an enzyme and typically an enzyme that is like degrading something or breaking the thing that it's after. And so this is an enzyme that's going to break the beta-lactam ring. And so when this happens, now this is going to break the ring, but it's going to break it in kind of differently from the way that the transpeptidase does it. What I mean by that is that beta-lactamase is not going to get permanently stuck. It's going to break open the ring of the antibiotic, but then it's going to be free to do things again. It's not like it's getting stuck on here. It's just breaking this open. And so it's inactivating your antibiotic. When you have the antibiotic inactivated, now the cell walls can be made, the chains can be linked, and the walls can be strengthened, so you can get resistance. If you don't have the antibiotic, then you can't make those strong cell walls, and so the cells can burst and die. So the BLA gene allows us to select for cells that have taken in the plasmid. But there are some caveats or issues that you need to worry about when you're using beta-lactamase, especially if you're using ampicillin. So ampicillin is subject to degradation, and so it can degrade fairly easily. So you want to add it very fresh to your media, so to the bacteria food. Add it right before you're going to use it, and be cautious if you're growing cultures for a long time that the antibiotic might be degrading. And remember, if the antibiotic degrades, then all the cells can grow, whether or not they have the plasmid. An alternative is carbenicillin. So it's more resistant to degradation, but it's also more expensive. But if you need to grow for a longer time or that sort of thing, it can be a good option. Another thing you need to worry about when it comes to this beta-lactamase is that it gets secreted out of the cells. So the cells kind of go on the offensive and they secrete this beta-lactamase outside of themselves in order to degrade the antibiotic before it can even get into the cells. And this can lead to shared resistance. So because it's getting secreted outside of where the cell is living, when you're doing this on a plate, you want these colonies to be nice and big, and sometimes what you see is you get these little tiny colonies around the colonies, outside of like the bigger colonies. And what we call these satellite colonies. And often these are little colonies that don't have the plasmid because they don't need it, because they're relying on the secreted antibiotic from the cells. So this is something you need to worry about when you're using these when you're using these penicillin, this ampicillin, these type of antibiotics, is to watch out for the satellite colonies. And you can reduce this chance of this happening as well by growing for a shorter time period. Speaking of time periods, when you are doing the antibiotic selection with ampicillin and other beta-lactams, because you're targeting cell wall synthesis rather than protein production, you don't need to have an outgrowth step, or at least you don't have to have as long of an outgrowth step. So what I mean by this is that when we stick the gene into the cell, so remember, this is a process called transformation. And so we often do it with this process called heat shock, where we take these cells and we take these bacterial cells, we like chemically weaken them, and then we heat them up, and then the plasmid is going to come in, and then the cells kind of heal themselves and they start making proteins, and we let them do this in the absence of antibiotics in this outgrowth step, and then we let them grow on, and then we plate them on the cells with the media that has the antibiotic. This outgrowth step is going to be really important if you have an antibiotic that is going to target protein synthesis. This is because you don't want to prevent protein synthesis before they can make the proteins that they need in order to inhibit the protein synthesis. So the plasmid is going to have the instructions for making the protein that's going to inhibit the antibiotic. And if we don't allow them to make those proteins, then we are going to not be able to make the thing that we need to inhibit the inhibition. So we typically do an outgrowth step, and based on the various cell types and the various antibiotics, is often like 30 minutes to an hour. And but with you're using the beta-lactam antibiotics, because you're not targeting protein synthesis, just the cell wall synthesis, then this outgrowth step isn't as important, but it can still help. And if you need to like increase the efficiency to allow the cells to heal a little. So that was the cell wall inhibition with the ampicillin and other beta-lactams. Now let's talk about the translational inhibition. There are several different classes of antibiotics that target protein making, but in different ways. So if we think about protein making, so if we think about translation, this is the process in which messenger RNA, so the RNA copy of a gene, is translated into protein. So you have these ribosomal complexes. These are made up of proteins and RNA. They're going to travel along the messenger RNA, and they're going to be reading out the RNA's instructions. tRNAs, so transfer RNAs, with the corresponding amino acid, are going to bring that amino acid to the ribosome, and the ribosome is going to add it to the chain based on so these tRNAs have a matching anticodon to the codon in the messenger RNA. The important thing here is that the ribosome is going to be smart, and so it has this proofreading ability that it's only going to allow the correct transfer RNA, so the ones with the right anticodon and the right amino acid, to bind, and it's going to prevent the wrong tRNA from binding in normal cases. And it's going to keep, when these bind, it's going to help catalyze the transfer of to form this peptide bond, and then things shift over, and then you have another one come in, et cetera, to form a functional protein. And different antibiotics can bind to different places in the ribosome and prevent this action. So in a ribosome, this is a bacterial ribosome, you have a 50S subunit, this big one, this large subunit, and then a 30S subunit, the smaller subunit. With these three sites, so you have it's enter in the A site, the chain is held in the P site, and then the exits in the E site, and then you have kind of like this tunnel where it's coming out. So there are a bunch of different places that antibiotics can bind and inhibit with this process. And so we see different antibiotics bind to different places and inhibit different steps. So let's start with the tetracycline antibiotics. So examples of this include tetracycline, chlorotetracycline, oxytetracycline, doxycycline, and minocycline. So cycline, okay, well, it has all these cycles. These are polyketides. This is a ketone where you have a carbon bound to an oxygen with carbon-containing groups on either side. And so multiple, and so tetracycline antibiotics have multiple ketones. What these do is they're going to bind to the 30S subunit, so that smaller subunit, and prevents the binding of the tRNAs. So even if it's the right tRNA, it's not going to bind. And if the tRNAs can't bind, then you can't make protein. And so the protein can't, they can't multiply and that sort of thing. Just a sort of medical side note is that tetracycline antibiotics, they can also bind metals like calcium, and therefore they can get incorporated into growing bones, making them yellowish. And they also make it harder for your body to absorb. So they're often not used as like a first line treatment, or used in people that are growing or pregnant or various things like that. But I'm not going to get into the medical stuff, except that the only reason I really know this is because when you make the tetracycline stock, it's like this yellowish color, and it got me curious. Okay, so that was the tetracycline, how it works. How do we prevent it from working? So antibiotic resistance can be given through having this TET-A gene. This TET-A gene is going to make this efflux pump. So efflux meaning it's sending things out. Influx would go in, efflux would go out. And so what it's going to do is it's going to pump the antibiotic out of the cell before it has a chance to start messing with ribosome stuff. And so it's not going to modify it, but it is going to pump it out, and this is going to allow the cells to keep growing. Another antibiotic that targets the ribosome, but in a different way, is kanamycin. So once again, kanamycin is going to be binding in that 30S subunit, but here it's going to be interfering with quality control. So it's not preventing tRNAs from binding, but it's not doing a good job checking whether those are actually the right tRNAs. So you can get misincorporation of amino acids. And if you're just allowing, if you're not too picky with what amino acids you're letting in, you can end up with gibberish proteins that are dysfunctional, and this can kill the cells. So how do you get resistance? Plasmids can get resistance by having a gene that modifies the drug. So instead of pumping it out, we're going to change it in a way that's going to prevent it from binding to the ribosome. So kanamycin, though it's drawn as neutral here, it's a aminoglycoside. So it has these amino groups and these glycans, so these sugar. These amino groups can be positively charged. They're shown neutral here, but they're typically positively charged. And in fact, we buy kanamycin as kanamycin sulfate, and the sulfate is going to be the negatively charged counter ion that's going to allow us to have this neutral salt of kanamycin sulfate. And then when you dissolve it, you get this positively charged kanamycin and the negatively charged sulfate ion, and they just can kind of like hang out because they have opposite charges. But the key thing is that because it's going to be positively charged, the ribosome is actually made up a lot of RNA in addition to some proteins. And so the RNA is going to have a negatively charged backbone, and this is going to make it attractive to the positively charged kanamycin. If you add a negatively charged thing to kanamycin, it's going to make it not want to bind as much, right? And so this protein, aminoglycoside phosphotransferase, can add this phosphate group. So it can add this negatively charged phosphate group to kanamycin to prevent it from binding to the ribosome. And if it can't bind to the ribosome, it can't mess up what the ribosome's doing. And so the cells will survive. There's a number of different names for the genes that encode an aminoglycoside phosphotransferase. So these include like CAN-R, NEO, and MPT2. And then the different aminoglycosides include streptomycin, gentamicin, neomycin, etc. And often these antibiotics, like resistance genes and stuff, will be compatible with no matter which of these antibiotics you use, kind of similar to how we could use ampicillin or carbenicillin. So they have different things with different price and stabilities and various things like that. So just to compare those two again, so kanamycin is going to make it so that it's less picky when it comes to what tRNA is binding. So you get dysfunctional proteins made. And you can give resistance to it by modifying the drug so that it can't bind. With tetracycline, again, you're binding to the ribosome's 30S subunit, but here you're actually preventing the incorporation of, you're preventing the binding of the tRNAs. And if the tRNAs can't bind, then no proteins can get made. But you can prevent this prevention by pumping the drug out before it can do its harm. And comparing this to ampicillin, so when we saw with kanamycin, here we're not pumping the drug out. So in addition to having the difference in that this is targeting cell walls, this is targeting protein synthesis, we also have this difference that with kanamycin, we're not pumping out the drug. With Betalactam, we're not pumping out the antibiotic resistance product, and similar with like tetracycline. With ampicillin, however, we are. And so this means that with ampicillin, we're going to have more of the risk of those satellite colonies than we have with kanamycin. So this is just something to keep in mind, is why you need to be a lot more cautious when you're looking, with how long you allow cells to grow, how suspicious you should be of little colonies and that sort of thing when it comes to ampicillin. Okay, the last example of an antibiotic that I want to tell you about is chloramphenicol, and I saved it for last because there's some cool stuff to say about it, in addition to its antibiotic selection role. So chloramphenicol, this is what it looks like. A note about it is you need to dissolve it in ethanol. The other ones we dissolve in water, this one you dissolve in ethanol. And speaking of dissolving, we typically make like 1,000x stock. This is very convenient. And then if you want to make like a liter worth, you add 1 ml. If you want to make 15 mils, you add 15 microliters, that sort of thing. And you can store like 1 microliter aliquots, or based on how much you normally use, just like make aliquots. So you make up the solution. I typically syringe filter them. So you take a little filter thing and stick it on the end of the syringe and you just put it through, and then you make aliquots. And based on the different antibiotics will be used at different final concentrations. So look up in the handbook or whatever that comes with it and says how much you should use, and then make 1,000 times that. Okay, so how does chloramphenicol work? Once again, we're disrupting the translation, but in this case, we're binding to the 50S subunit, so the bigger one, and we're going to be preventing peptide bond formation. In the process of translation, we have to have this peptide bond formation. We have to have this one peptide attached to the growing chain, and then this is going to get here, and then you transfer over. So it's really important that we're able to make this peptide bond, and chloramphenicol is going to prevent this bond from being made. If it can't get made, then you can't get proteins made, you can't get the cells to grow and copy. But something cool is actually happens that we can take advantage with chloramphenicol, a use that is not antibiotic selection. And so in this case, what happens is that we can use it to increase the copy number of a plasmid. So certain plasmids, so the plasmid copy number is basically the plasmids can have different copy numbers. So you can have different numbers of copies of that plasmid hanging out inside of the bacterial cell. So some plasmids, or in some cells types, you have like a low copy number where you only have like 1 or 2 plasmid copies. Others are high copy numbers, so you have a lot. And these are really good if you're doing things like trying to get a lot of DNA made. There are some plasmids that are under like stringent control about how many copies they have, and some that are under more like relaxed or loose control. If you have one of these plasmids with the PBM1 or COL-E1 origin of replication to this ORI, such as those in the PUC, PGEM, and PBR families, what you can do is this cool trick where you can add kanamycin and get a low copy number to become high copy number. The reason why you can do this is because they're under that relaxed control, which means that they don't need to make new proteins in order to copy that DNA. And so they need to make proteins in order to do all of the other stuff that they need to grow and survive and make proteins, but they don't need to make proteins in order to copy the DNA. They can just use the machinery that's already there. And so you can get them to make lots of copies of the DNA. without allowing them to be wasting all their energy making proteins and other things. So that's a cool trick, but of course it's not going to work if your cell has resistance to chloramphenicol. So how do cells get resistance to chloramphenicol? Resistance can be generated by chloramphenicol acetyltransferase, so CAT, and it's going to inactivate chloramphenicol by acetylating it, which is going to prevent it from binding to the ribosome by adding an acetyl group onto it. What's really, really cool about chloramphenicol is that it has this sequence-specific property where it's only going to inhibit at certain sequences. So it's going to cause the ribosome to stall at specific sequences. You can imagine that as the ribosome, as the peptide, the growing peptide is leaving the ribosome, you kind of have this site. And so we talked about this a little when I was talking about enzyme inhibition, because it's this really cool instance of a uncompetitive inhibitor. So basically it's going to only bind to the enzyme when the enzyme is also bound to the substrate. And in this case, it's only going to bind when there's a certain amino acid in a certain sequence leaving this tunnel. And so this allows it to stall at specific sequences. Bacteria take advantage of this like bacteria with resistance to chloramphenicol. So bacteria that have these resistance genes, they have this really cool mechanism where they have this other gene, this other sequence up here. What happens is the ribosome starts making this, but then it has the sequence that's going to cause it to stall if chloramphenicol is present. So if chloramphenicol is present, then that will stall, and this will stall in a way that's going to change the messenger RNA structure so that the gene that has the instructions for making the resistance gene, the resistance product, so that has the instructions for making the acetyltransferase, is going to get expressed. So normally it wouldn't, because that sequence would be hidden, but when you have this stall, then this is going to expose that sequence so you can get the these genes made. And when these genes made, they're going to modify the antibiotic, and they're going to modify it in a way that's going to prevent it from binding. So some really, really cool work and some really neat papers that came out recently about context-specific antibiotics and the structural basis for it. So chloramphenicol is just one example of a sequence-specific antibiotic. There are others like oxazolidinone, and so this is the structure here. And so there's really great structural work, these pair of articles that came out recently that I will provide links to. And there are other drugs as well that work in similar mechanisms and similar things. And I have to say, I'm a little biased because I'm going to be joining the Fujimori lab a week from Monday for my postdoc, and I'm incredibly excited, and I find this recent work really fascinating. So we can take advantage of these antibiotic resistance genes, but it's also important to remember that these antibiotic resistance genes are coming because they arose in the wild in bacteria, and antibiotic resistance bacteria is a very big growing problem, and we need to have more investment in it. It might not be as exciting of a topic in terms of getting money for that sort of thing, but it's this really, really crucial problem that is facing us as we have a growing antibiotic resistance bacteria problem. And so that's just a little plug for more research and more funding into this sort of research.